your work. So now it's the turn for Dr. Kenji ESHRE. He's a senior scientist at the Research and Development Laboratory of the Kato Ladies Clinic in Tokyo, Japan. Kato Ladies Clinic is the largest IVF clinic in the world. He obtained his PhD degree in agriculture from the Tokyo University of Agriculture and Technology. His research interests include fertilization, embryology, cryopreservation, and blastocyst implantation. Dr. ESHRE has participated in an extensive number of scientific publications in high-impact journals. Today, Kenji will deliver his presentation titled Unlocking Better Vitrification Outcomes, and if the fatty acid supplementation is the key or not. So please, we are ready when you are.
Thank you very much. And good afternoon, everyone. So in this lecture, I will talk about clinical effectiveness of fatty acid supplemented warming solutions. So as you know, cryopreservation technology for human oocyte and embryo has substantially progressed in the last 2 decades. So recent paper reported the actual outcomes of autologous oocyte vitrification, and the survival rate was reported as 78%. So this rate was higher than the competency value, but lower than the benchmark values proposed by Alpha Consensus in 2012, which sets a benchmark value at 85%. Furthermore, previous studies reported that oocyte vitrification reduces developmental competence and live birth rate. Furthermore, oocyte vitrification adversely affects the distribution and the function of mitochondria, chromosomal segregation, and zonoperucid properties. So from these results, we consider that there is a room for improvement in the vitrification techniques, and some modification to solutions or protocols are necessary to improve the clinical outcomes. So to devise a strategy, we consider the principle of vitrification and warming. So during vitrification, equilibration of cells with cryoprotectants are necessary to prevent ice crystal formation. So ES contains EG and DMSO as a cryoprotectant, which diffuse into the cells. And next step is BS, which contains higher concentration of EG and DMSO, and also contains non-permeable cryoprotectants, sucrose or trehalose, resulting in cell volume shrinkage due to dehydration. And in similar to ES, EG and DMSO diffuse into the cells. And then warming process. So this process involves 2 critical steps. So first one is removal of permeable cryoprotectants, and the second is rehydration. So at this time, I'm focusing on the first one, so removal of permeable cryoprotectants. So TS is a liquid with the highest concentration of non-permeable cryoprotectant, which expels the cryoprotectant from the cells. And in DS and WS, permeable cryoprotectants diffuse out from the cells. To improve the vitrification outcomes, we have focused on the one of the permeable cryoprotectant, DMSO. As an amphiphilic molecule, DMSO may influence the intracytoplasmic lipid droplet during vitrification and warming. Therefore, we previously examined the impact of vitrification on the lipid droplet in our plasma. So this red fluorescence indicates a lipid droplet in cytoplasm. So our data show that oocyte vitrification significantly decreases the lipid droplet in cytoplasm. So lipid droplets serve as a storage for lipid, such as triacylglycerol, TAZ. And the TAZ are broken down into fatty acid and then transported to mitochondria. Through beta oxidation, fatty acids contribute to ATP production and the subsequent embryo development. Therefore, loss of lipid droplet might be one of the reasons for reduced developmental competence. And we also hypothesize that compensating for lipid droplet may improve the embryonic outcomes. So we first speculate of how lipid droplet decrease during vitrification and warming. And we hypothesize that the reduction of lipid droplet occurred when DMSO is removed from the cytoplasm. We also hypothesized that adding fatty acid to warming solution may increase the fatty acid and lipid droplet in cytoplasm. So, to examine whether our hypothesis is correct or not, so we did some experiments. So first, we examined whether adding fatty acid to warming solution could restore lipid droplet in cytoplasm. And our data show that adding fatty acid to warming solution significantly increases intracytoplasmic lipid droplet. We then examined the developmental competence of these oocytes by ESHRE, and the developmental rate to blastocyst and expanded blastocyst stages were significantly improved by using fatty acid supplemented warming solutions. So from these results, we consider that adding fatty acid into warming solution effectively improved outcomes after oocyte vitrification. And then we examined the effectiveness of this solution for embryo warming. So we warmed mouse fossil stage embryo with or without fatty acid, and then transferred them to the oviduct. And our data show that fatty acid supplemented warming solution significantly improved the pup rate. Furthermore, we warmed bovine fossil stage embryo and cultured them, and the developmental rate to blastocyst and expanded blastocyst stages were significantly improved by using these solutions. So these results demonstrated that fatty acid supplemented solutions are effective for embryo warming as well. So our next question is, are the fatty acid supplemented warming solutions effective for human embryos? So to answer this question, we did some experiments. So this slide presents the outline of in vitro experiment using human cleavage stage embryos. So these embryos were vitrified at the fossil stage on day 2. And then embryos were randomly assigned to 2 groups. And we warmed and cultured the embryos to assess developmental competence. Additionally, to evaluate the blastocyst adhesion and outgrowth, the blastocysts were plated on fibronectin-coated dishes after donor pellucid removal. So this graph demonstrates the developmental rate to each stage. So although the developmental rate to expanded blastocyst stages are tended to be higher in fatty acid supplemented solution group, however, there was no significant difference. However, morphological grade of blastocyst was significantly improved in the fatty acid supplemented group. And this slide shows the outgrowth outcomes. So although adhesion rate were comparable between the groups, outgrowth area was significantly increased in the fatty acid supplemented group. So these results suggest that the fatty acid supplemented solution may improve the pregnancy outcomes after frozen cleavage stage embryo transfers. So we compare the pregnancy outcomes after frozen cleavage stage embryo transfers. The characteristics of patients were comparable between the groups. And our data show that the rates of implantation, clinical pregnancy, ongoing pregnancy, and rebirth were significantly improved by using fatty acid supplemented solutions. The rebirth rate was increased by 7% by using fatty acid supplemented solutions. And also, there are no significant difference in the maternal and perinatal outcomes. So our data demonstrates the effectiveness and the safety of fatty acid supplemented solutions. We then assess the effectiveness of these solutions for blastocyst warming. So we evaluated blastocyst viability using an in vitro outgrowth model. The adhesion rate, outgrowth area, and outgrowth degeneration rate were analyzed. The embryos were classified as degenerated when the outgrowth area decreased by more than 10% compared to the maximum outgrowth area. And there was no significant difference in adhesion rate between the groups. However, outgrowth area was significantly increased in the fatty acid supplemented group. Furthermore, blastocyst degeneration rate was significantly reduced by using fatty acid supplemented group. We then compared the pregnancy outcomes after frozen blastocyst transfer between fatty acid supplemented solution and non-supplemented solutions. The characteristics of patient and embryos were similar between the groups. And our data demonstrated that this solution can improve the pregnancy outcomes. And the survival rate was increased by 4% in the fatty acid supplemented group. So, taken together, our data demonstrated that vitrification decreases lipid droplet in cytoplasm. However, adding fatty acid to warming solution can offset this effect. Additionally, the pregnancy outcomes were improved by fatty acid supplemented solution without any adverse effect on maternal and perinatal outcomes. Therefore, we consider that use of fatty acid supplemented solution is an effective option for enhancing clinical outcomes. So thank you very much for your kind attention.
Thank you very much, Kenji. Well, congratulations to all three of you on the fantastic presentation. So now is